Good morning. We are going to start a lecture on secondary metabolites and secondary metabolism. Your course, Introduction to Ecological Chemistry, I offered several lectures, and it is better to have first midterm in next week. So next Thursday, you shall have uh, first midterm. So, Rahat, please let me know what uh, till today what we already been studied. Then I shall make question according to the your studied topics. So
Sir,
your
I will let you know.
yes, you shall have first midterm on Thursday next week. In our last class, we discussed on the function of secondary metabolites. I discussed in detail, and I hope you understood well. And if you have any question, you can ask me today. If no question, then I shall move second slide. Secondary metabolites and their proposed function. Here are some examples. For example, rutin. Rutin is a flavonoid. Here you can see it is a visual pollinator attractant. In the coloring plants or flowers, rutin is present and it contains lots of hydroxyl groups. This is why this is a powerful antioxidant and medicinally very important. So if you take red amaranth, that means lal shak, and other colorful vegetables, you can get lots of routine, and routine has anti-cancer principle also. Another interesting compound is you see rotenone. Rotenone is a powerful anti-feedant compound. Uh, that means insect feeding deterrent. And this rotenone has already been used in the practical field as fish poison. Other compounds like linalol, olfactory pollinator attractant, very simple compound. Barberin, this is another plant secondary metabolites. It is a defense toxin, toxic for the insect and herbivore. Similar toxin is dimbua, very simple structure, and brasilexin, antifungal compound. These are some examples. There are lots of examples of defense as well as other functions of secondary metabolites. In the Journal of Chemical Ecology, there is a journal named Journal of Chemical Ecology. Uh, it is dedicated to publish the research discoveries on chemical ecology. There are many other journals just like uh, Journal of Natural Products, Phytochemistry, Organic Chemistry, Organic Letters. They also published new discovery in secondary metabolites and their ecological role. So, production of secondary metabolites for defense against herbivores and pathogens is not necessarily constitutive. This is very important point. Constitutive. Constitutive means when you analyze a plant, you can find that compound as a constituent. Uh, yeah, that means plant carries that compound, biosynthesize and carries in their cell. But there are some compounds during wounding and infection, uh, they are triggered to induced accumulation. Uh, that means they are not, you know, present as constitutive compound in the plant cell. But when plants uh, have wounding somehow, be wind or storm or any other, or insect biting, or any herbivore biting, if there is wounding, or if a microorganism infect the plant, then plant can sense and induce to produce some secondary metabolites. They are called induced accumulation of secondary metabolites. So plant wounding and infection, for example, wounding can be happen through herbivore or other mechanical injury, or infection can be happen by different kind of microbes. In that case, secondary metabolites are biosynthesized. For example, in case of herbivore, to stop herbivore to uh, eat the plant, activation of prefabricated defense chemical. That means previously that compound present but not in the toxin form. So that constitutive compound can be transformed into a very toxic compound to stop the eating by the herbivore. Increase of existing defense compounds. For example, a toxin is there, but dose is concentration is low. So existing defense compounds, the amount or concentration can be increased so that it can stop the herbivore to eat the plant. Other way is induction of de novo synthesis of defense compounds. They are called phytoallergens. So phytoallergens are not constitutive secondary metabolites, but when plants are infected by microbe or some other herbivore, they are attacked. In that case, they can synthesize new compounds. De novo means inside the cell through a defined biosynthetic pathway, and they are, basically antimicrobial and antioxidant compound, and they are known as phytoallergen. Phytoallergen, several hundred phytoallergens have been discovered in plants, and this is really amazing, system in plant. So if somehow you can induce plants to produce phytoallergens, then you can, it is like vaccine. For example, in our case, we are going to have COVID vaccine, COVID-19 vaccine. Uh, in that vaccine, mRNA, mRNA of the virus will be, introduced, synthetic mRNA, and that mRNA will produce uh, the spike protein in our cell. And once our cell can sense the presence of spike protein, uh, cell realize that COVID-19 virus is there, and immediately they produce antibody, T cell, and other defense system to uh, stop the viral, attack. But actually, it is not virus, only spike protein. Spike protein, the job is induction of antibody or defense system. So in case of plant, phytoallergen is like, antibody or defense, induced defense system. So this is very interesting subject in chemical ecology, and scientists found that accelerated production of phytoallergen during during infection by the microbes and herbivores. So here you can see one example: the mustard oil bomb, a binary glucosinolate myrosinase chemical defense system. You know all mustard plant. in our country, it looks very nice when at the field flowering stage. And mustard plant, if you touch, or mustard oil, if you have, obviously you tested, you can find the pungent test or pungency, strong, uh, uh, aromatic compounds are there. Basically, mustard plant contains glucosinolates. Glucosinolates, they are not toxic at all, and they are present in the leaves. But nearby there is a myrosinase enzyme. So when plants are attacked by aphid or any other way, you can see mustard leaf. If you break down it, you can find strong aroma, and that are sulfur, elemental sulfur. That means immediately during injury uh, or damage, myrosinase enzyme activity, uh, you know, uh, increase, and myrosinase enzyme becomes activated, and it break down the glucosinolate, the actually uh, inactive compound. But glucosinolate, this is a mustard oil bomb. This compound, by the action of myrosinase in the cell, it breaks down, and it produces isothiocyanates. You know, cyanate is very toxic compound, nitriles and elemental sulfur. So elemental sulfur, the order you can find, thiocyanates, oxazolidine thiones, and epithionitrile, and these five compounds are produced. Here you can see all these five compounds, and they are basically toxic against the insect. And this is really a fascinating, the defense system in the mustard plant. uh, when mustard plants are affected by any insects or other herbivores, then myrosinase comes in contact with the glucosinolates, and the breakdown products I mentioned, these five, and they are toxic compounds for the herbivore. And this is an important, mechanism how chemical defense system, glucosinolate, myrosinase, a binary system help mustard plant to protect themselves from the attack of most of the insects and herbivores. Uh, Similar mechanisms are also present in other plants. So I am giving you an example. So today I would like to take a brief, the quiz exam uh, until today's lecture. So what you need to do, write 10 keywords uh, uh, in your subject, uh, introduction to ecological chemistry, and send me through messenger, Facebook messenger. So what you need to do, just type 10 keywords that you have never been heard or learned from any other subject. This is very simple topic. So only you learn from this course BTL 5 to 7 through my lectures. So identify 5 keywords, for example, glucosinolates. So you cannot write because I have given the example glucosinolates. It is a keyword, or keyword maybe 2 words or 3 words, does not matter. So write 10 keywords and send me within 5 minutes through my Facebook Messenger. And that's all for today's lecture. Do you have any question?
Sir, Sir, I didn't hear what you said due to network problem. Could you please repeat?
Yes, write 10 keywords that you have learned for the first time in this course, Introduction to Ecological Chemistry. You can do from your mobile phone also and send me through Facebook Messenger. And then I shall consider it as a quiz exam for you. Clear? Do anyone have any question? Rahat, is it clear?
Yes, sir. Yes, sir.
Okay, Fatima. Kanis Fatima, is it clear? Shafi clear?
Sir.
Joy Devnath?
Yes, sir. Clear.
Kanis Fatima, unmute yourself and then tell us whether you heard the question or not. I am giving you in the chat box. Write 10 keywords that you have learned for the first time through my lectures on BT on the course BTL 5 to 7 and send me through Facebook Messenger. So your time is started from now, and after 5 minutes I shall thank you so much.